Hello, my name is Mark Shepard, and I'm excited to discuss the role of connectivity in shaping latitudinal biodiversity patterns in marine ecosystems. While we often think of biodiversity in terms of climate or habitat complexity, you might be surprised to find out that connectivity plays an important role in shaping species distributions and genetic diversity in the ocean. Understanding this concept is going to be really important to improving marine conservation efforts and predicting how species will respond to environmental changes. First, let's start with this idea of latitudinal diversity gradient. This is a well-known and documented pattern where biodiversity is highest in the tropics and declines as you move towards the poles. There have been many traditional explanations as to why this latitudinal diversity gradient exists. One is energy availability. In the tropics, there's more sunlight, and warmer temperatures promote primary productivity, supporting larger food webs. A second reason that's been proffered is habitat complexity. If you take, for example, coral reefs in tropical ecosystems, they provide more ecological niches, allowing more species to coexist. A third one is evolutionary time. This proposes that the tropics have been geologically stable for longer periods of time, allowing more time for speciation to occur. However, another key factor shaping biodiversity in the oceans is connectivity. What this means is how species move, interact, and exchange genetic material across space. Connectivity can help maintain biodiversity by enabling gene flow, but it can also limit biodiversity by reducing opportunities for species to evolve in isolation. Today, I'm going to explore how connectivity influences marine biodiversity and why it plays a different role in the ocean when compared to terrestrial ecosystems. In the ocean, connectivity is largely driven by currents and dispersal mechanisms. Unlike land-based ecosystems, where mountains or deserts create barriers, the marine environment is more fluid. This means that tropical regions will experience high connectivity with strong gene flow between populations, leading to species mixing and genetic diversity. Polar regions are more isolated, meaning species that reach these areas tend to evolve independently, resulting in unique adaptations and higher rates of endemism. Depth also plays a major role in connectivity. The surface ocean is well mixed by currents, while deeper layers are more isolated, creating distinct biological communities. For example, diatoms, a type of microscopic phytoplankton, show varying connectivity depending on ocean currents and depth. In surface water, species like the Lysioria are widely dispersed by equatorial currents, leading to high genetic mixing. However, deep-dwelling diatoms, such as those found in polar upwelling zones, often experience more isolation due to differences in water density, temperature, and nutrient availability. This contrast shows how both horizontal and vertical connectivity influence biodiversity patterns. Additionally, seasonality plays a role in connectivity, especially in the higher latitudes. In temperate and polar regions, seasonal changes in temperature, ice cover, and ocean currents influence migration patterns, breeding cycles, and larval transport. This means connectivity varies throughout the year, further shaping biodiversity distribution. It's important to recognize that connectivity plays a different role in marine versus terrestrial ecosystems. On land, species are often isolated by physical barriers like mountains, deserts, and rivers. This limited movement increases speciation rates. In the ocean, species can disperse over vast differences. Some areas, like the Indo-Pacific, act as biodiversity superhighways, while others, like the Southern Ocean, act as barriers to movement. A coral reef, for example, shows extreme biodiversity due to both connectivity and habitat complexity, while open ocean environments have much lower diversity despite high connectivity. Thus, even though the oceans cover 70% of the Earth's surface, there are actually more species on land than in the ocean. This is because land habitats are more fragmented, leading to faster speciation. In contrast, The ocean's vast interconnectedness allows species to mix freely, maintaining genetic diversity but reducing opportunities for isolated populations to evolve into entirely new species. Although the ocean appears vast and open, certain barriers still limit connectivity and influence biodiversity patterns. These barriers can be physical, chemical, or biological. Some of the major barriers include temperature and salinity gradients, deep sea environments, strong ocean currents, oxygen minimum zones, and geographic features such as mid-ocean ridges, trenches, and continental shelves that can act as natural barriers separating populations by depth and geography. For example, the Antarctic circumpolar current isolates many Antarctic species from warmer waters, while deep-sea hydrothermal vents are home to species that rarely mix with shallower communities. These barriers shape marine biodiversity by influencing where species can spread and evolve. While connectivity plays a major role in shaping biodiversity, there are some challenges to the idea that it's the primary driver of species richness, or even that latitudinal diversity gradients apply equally to all marine organisms. Some marine species don't follow the latitudinal diversity gradient pattern. Diatoms, one of the most diverse group of phytoplankton, show their highest species richness in colder, nutrient-rich waters at higher latitudes. So unlike corals and reef fish that thrive in warm, stable environments, diatoms depend on seasonal mixing and upwelling, which are strongest in temperate and polar waters. For example, the North Pacific and Southern Ocean host some of the highest diatom diversity because strong seasonal changes drive nutrient availability, supporting rapid species turnover and evolution. There's also evidence that too much connectivity can limit speciation. While connectivity promotes genetic diversity, it can also prevent species from becoming genetically distinct enough to evolve into new species. For example, highly mobile pelagic fish like tuna and swordfish show very low genetic differentiation across ocean basins because they mix too frequently for isolated populations to emerge. Another issue is physical and environmental barriers that restrict movement. Not all marine species can move freely across the ocean. Temperature gradients, salinity differences, and deep-sea barriers limit connectivity in ways that aren't always considered in latitudinal diversity gradient theories. For example, the Arctic and Antarctic ecosystems remain highly isolated due to extreme temperatures and unique current systems, leading to distinct evolutionary lineages. These challenges suggest that while connectivity is a powerful force in shaping marine biodiversity, it interacts with other factors such as nutrient availability, environmental stability, and isolation to create complex and sometimes unexpected biodiversity patterns. It's obvious that to fully understand connectivity's impact on marine biodiversity, we need more research, particularly in three important areas. One, genetic studies. So by comparing genetic diversity across latitudes and depths, we can track gene flow and understand how populations are connected. For example, how do tropical reef fish compare genetically to deep-sea species? Another area of research, ocean current modeling and dispersal studies. So for example, using satellite data and ocean models, we can map species dispersal routes and predict how currents influence migration. For instance, how do major currents like the Gulf Stream or equatorial currents affect larval transport? And then a third area, long-term biodiversity monitoring. To detect trends over time, we need continuous observations. As ocean temperatures and currents shift, do species ranges expand or contract in response? Additionally, more research is needed on how seasonality affects connectivity, particularly in higher latitudes where species dispersal fluctuates with changing ocean conditions. Understanding how connectivity varies throughout the year can help predict how species respond to climate change and other environmental shifts. So in summary, connectivity is a key driver of biodiversity in marine ecosystems, influencing where and how species are distributed. But there are some other things that are going in the ocean that influence biodiversity. For example, in highly connected regions, biodiversity can increase due to genetic exchange, but it also risks homogenation in other areas. Also remember that in isolated areas, species evolve independently, leading to higher rates of endemism. This is what you see in coral reefs. Also remember that not all species follow the latitudinal diversity gradient. Some, like diatoms, show peak diversity at higher latitudes due to nutrient availability and seasonality. Seasonality itself plays a major role in connectivity, particularly in higher latitudes where seasonal changes in temperature, ice cover, and currents impact dispersal patterns.